might be surprised to learn that mechanics is an important feature of our human biological systems. Mechanics explains how our blood clots, how our hearts beat, and our hair curls, among other processes. These and other mechanisms are the subject of study of Associate Professor of Mechanical Engineering, Malebogo Ngoepe. When COVID-19 arose, for instance, Associate Professor Malebogo Ngoepe and her team turned their focus to how the virus might affect the body's coagulation system. She is the director of the Center for Research in Computational and Applied Mechanics, and has held an Isolomso Fellowship at the Stellenbosch Institute for Advanced Study. And I'm sure we will enjoy hearing from Malebogo, and thereafter I'll come and close, and then we'll go out and enjoy dessert and the sun and the interaction with our young scholars. Over to you, Malebogo.
Good morning, everyone, and thank you very much, Professor Pakeng, for the invitation to speak here and for your attention this morning. I'm going to share a bit about my work in biomechanics, and specifically I'm going to be talking about two projects which are quite close to my heart, that in blood clotting and that in hair. If you hold on tight to the end, I swear there may be a connection between those two things, but I'll try and make the case to show you how. So let me begin by explaining what a blood clot is in brief. Often when we hear of blood clots, that sends a shiver of fear through us, but blood clotting is a mechanism that the body uses to protect the circulatory system. In simple terms, if you cut yourself, you want to be able to stop bleeding. However, there are a lot of diseases in which blood clotting goes out of control, and that's where we have thrombosis and other challenges. So if you look on the diagram, there are three main players in the blood clotting system. You have the composition of the blood, or what's in the actual blood, so how many proteins do you have, how many cells do you have, and are they able to interact with each other? You have the state of the blood vessel, which is there a cut or not? In some cases, or in healthy cases, you want clots to form only when you have a cut, but there are cases, of course, of disease where the clots just form spontaneously. And those two fields have been studied since the 1960s by biochemists as well as vascular biologists who have brought us a long way in our understanding. The final piece of the puzzle is blood flow, because of course these proteins are carried in flowing blood, and whether or not they're able to interact with each other for long periods of time will determine whether or not you form a clot. And blood flow is typically studied by mechanical engineers because the same equations of fluid mechanics that apply in oil and gas pipelines or in the pipes in your vehicle are the same equations at play here. And so that's why I got interested in this field. So we have specific diseases of interest. Blood clots aren't always bad. Sometimes they play good cop, sometimes they play bad cop. And so depending on the specific disease that you're focusing on, it'll depend on how clotting affects that system. One of the systems that we study are brain aneurysms, and all an aneurysm is a weakening of your blood vessel on the brain that then forms a balloon that's likely to pop. Now, what doctors try and do is to get a blood clot to form in that balloon, because if that they can make that happen, it acts as a natural barrier that then prevents the aneurysm from popping. But of course, some people clot too much. So we've developed techniques that enable us to predict blood clotting in aneurysms on a per-patient basis so that we can see if the treatment would result in a good clot or a bad clot. And then as we were going along developing our systems, COVID came along. And what was found in a lot of individuals who were dying of COVID was that when biopsies were carried out and the lungs were opened up, they were finding thousands of little microclots in the small vessels of the lung. And of course, those are really hard to see with an MRI scan or a CT scan. So we designed one of the first systems that was able to grow those very, very tiny clots in tiny, tiny, tiny microchannels and put them under a microscope so that we could see how the clotting mechanism was playing out. And I'll talk a bit about our methods, and then I'll show you some of those results. So we use a range of computational methods to develop our models. And if we start at the top on the left, typically what we'd get is we'd get a scan of the patient's brain with the aneurysm, and then we take that aneurysm and the surrounding blood vessels and reconstruct it into a virtual aneurysm on our computers that we can then model blood flow in. And then we go ahead and solve the equations of fluid flow as well as the equations of the biochemistry that gives rise to a clot. And you'll see in aneurysms, the shapes differ from individual to individual. So this is why we want to be able to do it on a per patient basis. If you go to the bottom row on the left, you'll see a very colorful image. Once we've solved those equations, we're able to get a sense of the quantities of various proteins, how fast the blood is flowing, and we're also able to predict where the clot is. Now, it's all great and well to do this in a virtual environment, but how on earth do we know it's true? How do we know that we haven't just created a clotting metaverse that doesn't actually necessarily make sense in real life? So we spend a lot of time developing and working with collaborators on experimental techniques that'll allow us to run these actual experiments, because the ethics of putting a camera in someone's head for a long time and hoping for a clot to form would be really tricky. So we have to develop alternative experimental methods to be able to get that data. If we focus on the top row first, those are the methods that we use for diseases which we can see with the naked eye. So aneurysms and blood vessels on the brain are quite big. So first, on the left, we need something to grow the clot in. So we typically print 3D geometries, either based on the patient's geometry themselves, or we can form what we think is representative. And then in the middle, we hook it up to a pump with flowing substances that represent blood and the various proteins. And then we're able to use high-speed cameras and lenses to capture the growth of the clot, as well as quantify the speeds of flow. On the bottom row, we have experimental methods that we use if we're working in very, very, very tiny spaces, like the case of the COVID clots. Because, of course, the size of the vessels on the lung are about the width of a hair, so it would be very hard to see what's going on inside. So the diagram on the left, which is a microfluidic chip, if you see, has very, very thin lines, and that's where our blood was flowing through. Again, we hook it up to a pump, put it under a microscope, and we're able to observe the growth of those clots in real time. And what kind of information are we able to get? The top row, again, is our brain aneurysms. The bottom is COVID-19. At the top, we just took sequential photographs at 20 seconds, 40 seconds, 60 seconds, 80 seconds, watching the clot growing in real time, and we're able to capture that. And then the middle diagram was a measurement of the experimental techniques where we could actually measure the speeds of flow. And then on the right, we could compare our computer models to physical reality and ensure that what we're predicting is actually true. In the bottom row, we had our experiments from COVID. And the first picture on the left with the red circle shows a normal blood clot. So that's just a normal blood clot grown from healthy blood. You can hardly see the clot. It's almost like there's nothing there. You move to the middle and you can suddenly see the COVID clot, which is that gray shaded area, which was almost filling up the entire channel. And that happened in about 90 seconds, which is way faster than how normal clots were forming. But what was an even more interesting observation was what happened at washing up time. So we like to wash out our channels when we're done with experiments before we discard of them. And what we found was that you couldn't get rid of the COVID clots. All the other clots would just wash out quite easily. But these ones were hanging on for dear life and were not going anywhere. And various other studies have since shown that what goes wrong is the dissolving of the clots, the COVID clots in the body. And that's what a lot of long COVID sufferers also grapple with, is the fact that their body just forms lots and lots and lots of clots that can't be dissolved. Now, the final picture on the right is a microscopic scan of the actual clot itself, which is an entanglement of lots and lots and lots of fibers. And I'd like you to hold on to that as I go and talk about another fiber, which is hair, because I'll hopefully be able to show you that there may be a connection when we get to the end. My other real area of interest is in human hair fibers, and this is work I've carried out with the Hair and Skin Research Laboratory, headed up by Professor Nonhlanhla Khumalo at the Health Sciences faculty. And hair has actually been studied in quite great detail. The only challenge is that most of the sample used have been straight, and all of the assumptions have been assumed to hold across the board. Now, anyone with curly hair would know that curly hair has a life of its own, and it has superpowers. Like, it can be short on one day, it can stretch out after you plait it, you throw it back in water, it's short again. And so it has these abilities to live a life of its own, sometimes at odds with what the owner wants. Now, what we found is that, firstly, if we just put hairs under a microscope and you cut them across, they look very different. So you'll see in the image on the far right, which is very circular, that's quite a straight hair. And as hair gets curlier and curlier, the shape becomes more elliptical. Now, what's fascinating about hair, and straight hair and curly hair in particular, is that it's made up of the exact same building blocks. However, rearranging a few of the components and depositing them in different quantities gives rise to radically different mechanical behavior. And from a biomaterials perspective, there's increasing interest in the use of discarded hair, whether it's animal hair or human hair, as a source material for biomaterials, which are lightweight, cost-effective, and relatively easy to biodegrade. But before we can exploit that or make use of our understanding, we need to understand what the fundamental thing is doing. So we went off and carried out mechanical tests on hair fibers, and that's just a really fancy way of saying we pulled the fibers until they broke within a sophisticated system where we could measure what changes were happening when we were pulling. And the graph that you have there is very exciting for material scientists. Every single material can have a graph like that, and it gives you tons of information about how the material behaves. What I want you to focus on is on the curly hair fiber, which is a solid line, and just look at the shape of the curve. Starting on the left, it's flat, and then it rises steeply, and then it becomes flat again. Whereas if you look at straight hair fibers, which is the dotted line, it just rises steeply and then flattens out. And that little big portion in the beginning, which is quite flat, carries tons and tons and tons of chemical information, which up until now has been ignored, and that's part of what gives curly hair its superpowers. But when we first saw that in the lab, we thought, maybe we're just seeing things. But then we carried out more and more tests on curly fibers from different people, and we were like, no, this is a real phenomenon. Is there another material that perhaps behaves this way, just to make sure that we're not just going off on a path? And tendon collagen, which is in abundance elsewhere in the body, also has some kind of crimp or curl, and equally also has that flat region in the beginning. So here's that picture of the stretching ability. If you start on the left, that's how the fibers start. You pull them, they straighten out. You throw them back in water, and they shorten again. And my goal for the next couple of years is to actually understand what that chemical mechanism is. Because if we can understand what it is, how we measure it, and how we alter it, we can then have a way of radically changing the mechanical properties of materials that we design quite easily through changing a small chemical mechanism and giving rise to radically different mechanical behavior, as is the case with straight and curly hair. And then coming back to the clots, on reading further, it seems that there's a similar mechanism at play that makes those clots resistant to dissolving, because they equally become quite curly and entangled with each other. So if we can look at the macroscopic case of hair, which is easier to see, study it and develop techniques for it, we can hopefully go back to that question of why those COVID clots wouldn't dissolve. And if we can manipulate that mechanism, we may provide a way of being able to stop them. As always, it takes a village to do good science, and we have lots and lots and lots of local partners, both at UCT and at other institutions. And on that long list, with names and brackets in particular, some of our students who come from further afield, who have been an integral part of developing this intellectual work. We have students from Zambia, the US, Nigeria, Botswana, and the DRC. And we also have international partners in Canada, China, where they're helping us develop a chip to diagnose clotting in long COVID. We have partners in Ethiopia, in Germany. We also have Namibia, New Zealand, and the United Kingdom, and we're deeply grateful for their cooperation and partnership in enabling us to do this work. Thank you very much.
Wow, thank you very much, Malebogo. Very interesting, and as you can see, international footprints in all of the work that we're doing. Very exciting, and much of the work that Amir, Elona, and Malebogo is doing is growing work, and they keep saying, we look forward to see what's coming out of this, and I'm sure we'll hear a lot from them. So thank you, Malebogo. Thank you, Amir. Thank you, Elona. Thank you not only for giving us such fascinating insights into the kinds of challenges UCT researchers are studying, but also for the results that they are achieving and the positive changes they are making in our world. An important feature of research is teamwork, and we saw it in the work each one of them is doing. Collaboration, cooperation, teamwork, and transdisciplinary approaches are key factors in cutting-edge research today. And one reason is simply that the problems we are trying to solve are complex and they are intersectional. No single branch of science can answer all the questions that we are facing. Hence, we see an engineer who dabbles in biology, and another engineer who gets into blood clots and how they relate to hair. But in fact, they are trained as engineers, and they work with people in other faculties. In the same vein, research and education institutions cannot work independently of other sectors in society. The work that you have heard about this morning has all been made possible because of strong partnerships and generous funding from a range of organizations all over the world. And many of you would have been able to recognize some of the funding agencies that enable our work. So thank you to all your countries for supporting the work that we're doing. UCT is not just the campus on the hill. We are not just our academics, we are not just our students, and we are not just our professional staff. We are also the communities across the southern suburbs, the CBD and waterfront, the Cape Flats and beyond, including our South African tuberculosis vaccine initiative in Worcester. We have GSB satellite offices in Philippi and as far away as Sandton. Other township offices provide research and treatment initiatives for health conditions like HIV/AIDS and TB, reproductive health screening and treatment for women, community outreach on safety, policing, and urban planning, and a wide-reaching support program in local schools. We also work in other parts of the continent, including the 3 oceans along our coastline, where we do important research on climate change and biodiversity. Our mission as a university is not just to be the best university in Africa, but to be the best for Africa. An important part of that mission is embracing the different communities and partners we work with, developing relationships that will contribute not only to the research, but also to government policy, education, and the development of technology, business, law, and social needs that contribute to economic growth and well-being. Wherever we go as UCT, our message is UCT is your university. It doesn't belong to only a small part of our society, but it belongs to everyone in South Africa, the continent, and the world. And I believe our mission is something we share with all of you and with the various interests you represent. The desire to see Africa developing into a continent that provides knowledge and other resources to serve the wider world, and that is growing economically. I hope we can find opportunities to do this together in new ways. And this, of course, brings us to the end of our formal program. Let me thank you for spending this time with us. I hope you found it interesting. I hope you found it instructive. I hope you were excited about our young people, our future leaders who shared their science. Please feel free to continue to chat with my colleagues, our future leaders, people who presented and those who didn't, and get to understand more of our work. And please leave with the thought of how you and UCT can work together to serve Africa. And feel free to contact us and we can engage. We are the top university on the continent, and so we can be your best partner. Thank you so much for joining us.